Welcome everyone, and thank you for joining us. Hopefully we can shed a little light on some of these thoughts around the use of CRISPR and genome engineering technologies today. So most of you, I think, actually live in this translational genomic space that is really the link between raw genome information and the application of that information in patients. We at Horizon are really dedicated to this area, and we have now become gene editing experts, having created hundreds of genome edited cell lines in over 70 different cell backgrounds, derived from about 17 different tissue lineages. At Horizon, we like to think that we're technology agnostic. Our motto really is to use the best tool for the job. In-house, we use a combination of 3 different gene editing technologies. We use recombinant AAV, which is our proprietary technology. It's a very high precision technology. It also has utility as a donor. We'll talk a little bit about that when used in combinations with nucleases. Zinc finger nuclease is another technology that we use in-house. This technology has good track record. We have licensed it in for use from Sigma. And then lastly, why I think most of us are here today, to talk a little bit more about the CRISPR Cas9 system. It's a very simple design system. It really is enabling, I think, for everyone. And so that's why we're going to make it the focus of today's discussion. CRISPR-Cas9 is an RNA-guided mechanism, and it has very simple design rules. We're going to cover some of those in the next few slides. Originally, as discovered, it was a 3-component system. The first component consists of the Cas9 protein. This is the protein that's responsible for cleaving the DNA. Then there are 2 RNA components, one called the CRISPR RNA and then the tracer or tracker RNA, and I'm not sure which one of those is actually the correct one, which often get put together now. For those of you that have been reading all of the publications, they can be constructed as a single RNA called a guide RNA, and that's what we'll refer to it in the rest of the presentation here. Cas9 comes in a couple of different flavors too. There is the wild-type version of the protein, which causes a double-strand break in the target. And then there's the what we call the Cas9 nickase, or D10A mutant is the most common one, which actually just causes a break or a nick in a single strand of the DNA. As I mentioned, it really does have simple design rules. What you're looking for is a target that has 23 nucleotides that end in the sequence NGG, with N being any one of the 4 bases. You can see here an example of a guide target sequence, which is 20 base pairs, followed by these 3 that constitute the PAM. One thing that's important to remember, if you're doing a guide RNA design for yourself, you don't actually include the PAM sequence, those 3 bases, in your guide. But what you do is you take that target sequence and you append that to the fixed scaffold that together really comprises that complete guide RNA. There's a recent paper out of Keith Jung's lab at Mass General that suggests that a 17 base pair target region actually can reduce some of the off-target potential that the guides have. You can expect to see an eligible PAM site about every 12 base pairs in the genome, but it really does depend on the sequence composition of the area you're looking at. There are several sources of guide RNA design available today, and Horizon, we have our own, and we'll talk a little bit about that later too. So on the surface, genome editing really does appear pretty simple. You identify a guide RNA target sequence in the genome, you order an oligo with that target sequence, you clone it into a guide RNA vector, and then you transfect cells with that guide RNA plus the Cas9 protein, and voila. Well, it's not really that simple. There are other things that you need to consider. In reality, there are probably 6 or 7 primary categories of consideration, and I've kind of broken them down here. We're going to go through each one of these one at a time and talk about some of the considerations within each topic. But to make it a little more interesting, let's kind of give this a try. Let's make an assumption that our experimental goal is to modify a single allele with a point mutation in the coding region of a gene, and we're going to use CRISPR. And of course, you want to be successful the first time. You don't want to waste time in the lab. You don't want to spend any more money than you have to. And hopefully you get that important publication without going too crazy. So here's the plan. The first consideration comes on the gene target specifics. So how many copies of your gene exist in your cell line? Most of us are using transformed human cell lines. There aren't a whole lot of those transformed human cell lines that actually come with normal copy number of 2. If you look at the karyotype down below, for many, many years, many people were using HeLa, and you can see that it's a real mess when it comes to some chromosomes. It's important to understand your cell line. Do you need to modify all of the alleles in your target gene? When you make this modification, do you expect it to affect the growth of those cells? So this is something that has been confounding for many of our clients. For example, when they find that the gene alterations that we're trying to introduce don't seem to be able to be isolated, and they later come and tell us, for example, that their experiments with shRNA showed that the viability of the cells was affected greatly. So these may seem obvious to most of you, but these are considerations, and there's a checklist that you really need to go down to make sure that you can do this. One of the other things you can consider is, would it be okay, for example, particularly with CRISPR, if you were to knock out one of the alleles while modifying the other one with the intended alteration? That is a result that you often get with CRISPR, and we'll talk a little bit later about why that's the case. So the second category, and this is probably the one that causes us the most trouble here at Horizon, is the suitability of the cell line that you're using. So first off, you need to get the DNA into the cell. So does your cell line transfect or electroporate well? You need to know that. The biggie is, can you single cell dilute your cell line and have it come back at reasonable growth levels? This probably trips up more experiments than anything else in this whole list that we'll talk about today. We go through quite a rigorous characterization procedure here at Horizon to look at cells to make sure that they are suitable, that we can get the DNA delivered into them, and then most importantly, that they will single cell dilute. If you look at the graphic there on the top, you can see, for example, when you single cell dilute, for one, you have to think about, am I actually getting a single cell into that well, or is it, as the top panel shows, perhaps a triplet that's kind of stuck together? That can really confound your screening later in the process, and it can take you a long time to try and separate all of these different artifacts that you see. The other thing that I think a lot of people don't really appreciate is the single cell dilution, and the fact that even though your cell line may grow like a weed right now in your T25s, when you put that into a single well and leave it all alone, it may not quite be so happy. So we have actually looked at a whole panel of different types of media formulations, additives that you can put in there, different seeding densities. You can see the panel on the bottom. Just gives you an example of taking one particularly hard cell line and trying a range of conditions to find the conditions that are most suitable for single cell dilution. Okay, now let's get to the more interesting part, guide RNA design. It's probably one of the most important things to consider when it comes to this is, what sequence source are you using? If you keep in mind that a single base pair change can mean the difference between success and failure with most of these technologies, and CRISPR is definitely part of that, it's important that you know what the target looks like in your cell line. We highly recommend that you sequence all of the alleles in your cell line. You know what it is that you're targeting. That one base pair or two base pairs can really make a difference in whether or not you're going to be able to make an effective change. We have an online tool that we use here at Horizon that is being developed in collaboration with Desktop Genetics. You see a little example of that output. There are other publicly available sources that give you some of this information as well. But what you need to do is identify potential guides, as we talked about a little bit earlier, in your sequence. And then you have to figure out how close are those guides to the particular modification that you're trying to make. Distance is important, particularly for something like a point mutation that you're trying to introduce. The closer your cut is to that change that you're making, the more effective the guide RNA will be. The other things you have to think about, and this is definitely something about CRISPR that we pay a lot of attention to, are what are the potential off-target sites. So the algorithm takes into account all of the sites that have obviously a perfect match, or a 1, 2, or 3, even up to 4 base pairs of mismatch elsewhere in the genome. And sometimes you can't avoid having a couple of mismatches existing somewhere else. That's very common. But then you need to know, are these in the coding region of a gene? Are they in the non-coding region? If it's in the non-coding region, is it in an area that's likely to be involved in regulation? These are all things that you need to look at. So one of the things that you can do to get around the specificity issue is to think about what version of Cas9 you'd like to use. So the Cas9 WildType protein induces a double-strand break, as we said earlier. It only requires a single guide, which is nice. Generally, it will have a very high efficiency of cleavage, although we'll talk a little bit about guide activity in a minute. It's particularly good for random indels. If you're looking to disrupt the sequence using NHEJ, where you don't need to control exactly what goes on, but you're fine with some bases getting deleted or added that disrupt that coding frame, then the wild type is actually very good. It can also be very good because it targets multiple alleles at the same time. But there are concerns about the off-target specificity. Because it's a single guide that has full activity, if it is having off-target binding effects, you will see those effects more pronounced than if you use something like the nickase. So the nickase, as we mentioned before, only nicks one strand. It therefore requires 2 guide RNAs if you're looking for activity levels that are somewhat similar to what the wild type would be. But the guide efficiency is really dictated by the efficiency of the weakest guide RNA that you're using of those pairs. So that's something that's good to know information about. But it does reduce the likelihood of off-target events, and that can be very, very important. So really, in summary, the wild type is good for knockouts or disruptions, or if you can't find 2 guides that are readily near each other. The nickase would be my choice most of the time, being probably safer from an off-target perspective. So here's just a little bit more information about designing a guide and when you're looking at the wild type versus the nickase. As I mentioned before, when you're using the wild type, you're going to get a double-strand break that's going to occur 3 base pairs upstream of the PAM site. When you use the nickase, you get this single-stranded nick, but you need to also be aware of the fact that if you're using the D10A mutant, at least, that this nick is going to come on the opposite strand from your guide design. So you have to keep that in mind. Data has suggested that nicks that provide a 5' overhang are the most efficient then for further modification. This is something that we've also found here at Horizon. So when you think about your guide design, you really want to place your top strand guide to the right of your bottom strand guide so that you achieve this 5' overhang. But then that begs the question, how important is this difference in the distance between the 2 nick sites? And again, published data suggests that there is maybe a limit of about 100 base pairs to maintain that activity. Something we've seen here suggests that really that 100 base pairs is not a limit at all, but it does depend a little bit on the efficiency of the guides and what type of donor you're using in the modification that you're trying to achieve. Okay, let's move on to guide RNA activity. So are all guides created equal? Clearly no. The answer is no. The prevailing wisdom is that in order to ensure that you obtain at least 1 good guide RNA with reasonable activity, say of greater than about 3%, that you need to test 5 distinct guides. In our guide RNA validation service, which Chris will outline a little later, we test 5 guides for each target to identify the most potent guide. Well, how do you measure that activity? Well, the surveyor or mismatch assay is the most common method used, but that assay itself can be problematic as well. It involves PCR primer design so that you get a very clean amplicon, and then you have to include the proper controls or it can be fairly misleading. What you see here, the data panel, is actually a result from our own HEK293 CRISPR-ready line. This is a line that constitutively expresses the Cas9 nickase, and Chris will tell you more about that when it's available. But you can see here, for example, using clone 1, when the 2 guides are provided with each other, there's almost none of the original top band left. So the guides can be very, very efficient at this cleavage reaction. But you need to understand what that activity is. It's very important because, for example, if you have a 10% cutting efficiency, then you would expect that you would find several clones modified in a single 96-well plate. But that relies that your single-cell dilution is very accurate and reproducible, and that all of the modified cells, for example, would be modified in the way that you're looking for. And that's not always the case. So safety would dictate that you might want to plate out more plates. All right, let's continue on. We're looking now at the donor design part of this. As I mentioned in our hypothetical model here, we're going to try and introduce a single base pair change, a single mutation into the coding region of a gene. So we've got a guide now, let's presume, with reasonable activity that's cutting at the site, very close to the site of the mutation. In order to get this mutation adopted by the cell, we have to provide a donor template for the repair at the site where the break is being introduced. So I would say, to read many of the papers out there, you think it's just as simple as ordering an oligo that has this desired change in it. But I would say it is actually a little bit more complicated than that. One of the things that those papers do is often talk about the efficiencies with which these changes occur in the cell, but they don't necessarily go to the trouble to isolate single-cell clones and grow those up. And so if you're not seeing, the 50%, the 70% types of frequencies that you see reported in the publications, rest assured that it's not you. Most of the time you aren't going to be seeing levels of activity that are nearly that high. But let's get back to the donor design here, for example. So one of the best strategies for actually achieving the incorporation of your donor is to alter the target sequence in that donor in order to prevent further cutting by CRISPR. Seems sort of obvious, but it's very important that you take other things into consideration when you do this. For example, if you're in the coding region, like our hypothetical example, you have codon substitutions that you have to consider. You have to think about splicing. Are the changes you're going to introduce affect splicing? And then how big a donor do you need? How many changes are you introducing? Where do you derive that donor from? I mentioned before that for targeting, every single base pair is actually very important. But I would say it's also important on the donor side of things that you try and introduce as few mismatches as possible in your donor in order to achieve the highest efficiencies. Something else that you can do is you can use selection, but you have to be careful there too, because now you're introducing another open reading frame. So let's get back to our example, and let's think about what are the possible outcomes for our situation. So the desired outcome here, if you think about it, is where you've got a mutant on one allele and the wild type preserved on the other allele. And you can see just from this array here that at best, really 1/4 of the total possible outcomes are going to be the desired outcome here. Now, you can bias that if you can modify the donor to be resistant to digestion by the guide. So that is obviously one of the most important factors in trying to do this type of design. In our collaboration with Desktop Genetics, we've implemented a donor module in the design process that allows us to look at the coding regions, for example, and as you can see on the lower right side here, take into consideration what codon changes can be introduced in that guide, where they will appear within the guide, and how those changes occur. One of the things that's important when you're thinking about codon substitutions is, are you substituting a codon of similar frequency? If you have a codon that is used highly and substitute it with one that is very rare, you could have a severe effect on the expression of your gene. So it's important to take that into effect. Not all codon substitutions really are the same. Another thing about the donor, I just want to bring this out for now, is that occasionally you can combine these technologies. And here, having our background in recombinant AAV, we've used that technology together with CRISPR and actually found that AAV can provide the most efficacious donor in many situations. Here you see an example of a cell line that was not particularly easy to transfect, but still we were able to get the CRISPR reagents in. And then you can see by using AAV as a donor, we're getting roughly a 50-fold increase in the uptake of that donor as opposed to either an oligo or a double-stranded plasmid. Now, back to our considerations. Screening. Okay, you've gotten this far, congratulations, but really this is where the hard part starts. How many cells should I plate? How many do I need to screen? Well, really the combination of transfection efficiency, guide cutting efficiency, the donor design, how well your single cells dilute, whether you've got a modified donor, all of these factors play into what sort of frequency you think you can expect to see coming out the other end of your experiments. So I think don't be misled by saying I've got a guide that has 10% cutting efficiency and then expect that you're going to see 10% efficiency in incorporation of this mutant oligo. That's just really not the case. But there are many factors that play in here, and it's very important to understand those. You need to be able to identify the modification that you're putting in there. So I suggest, and I think most people use, a PCR-based assay that can detect the presence of your modification in a pool of cells. You don't want to go through a whole bunch of single-cell dilution and growing these up for a couple of weeks to only find out then that probably nothing ever happened in your cell line. So it's nice to take some measure of the pool of your cells and be able to apply something like a PCR assay to see if at least the change that you're looking for exists somewhere in that pool. Then you've got to grow those cells up. You take a sample then, you screen that by PCR. But then you have to think about, are there multiple alleles that have been modified? Will your PCR strategy actually distinguish the wild type from the desired mutant from something like an indel, which is very common in CRISPR? You can use Sanger sequencing, but you have to be prepared for some messes. There's multiple alleles, there's mixed clonal populations. It can be very hard to tease all of this apart. And then you need a confirmatory strategy. It's probably best to use sequencing, and you need to look at enough isolates to confirm that you really do have all of the alleles covered. And then finally, validation. You have to think for your purposes. Do you need to be looking at the potential for off-target site modification? So most of the design algorithms give you an idea of where potential off-target sites are. Do you want to go look at those? You want to do some deep sequencing? These are all considerations, and it really depends on your particular application. Don't be surprised, too, if your modified allele isn't expressing. It does happen. This is something that we've stumbled across quite a few times. You need to be prepared to say not only have you made a genomic modification, but if the expression of that modification is important, you need to have a strategy in place to look at that. The stability of that allele over time may vary too, depending on the background of the line. It's always good to freeze some early passages if you can, so that's very important. And then there's always contamination. Let's not forget that some of the lessons that we've all learned from Gila, basically taking over the world. So, what is the real answer? Well, for those of you that are fans of science fiction, the answer is 42, but you already knew that. Actually, what it really boils down to is doing genome editing is a little bit more complicated than it might seem on the surface. So let Horizon help you. I think that's really what this kind of boils down to. We offer services and products that Chris is about to describe to help you achieve your gene editing goals. Chris?
Thank you, Eric. So I'm going to spend the next few slides just describing at a very high level what some of the products and services that we offer are, along with some of the ones that we already have on the market and others that are going to be coming very soon. I am going to cover these at a very high level, so I would encourage you to take a look at our website. We have a lot more detail there. But on this slide, you can see that everything we offer falls into 6 broad buckets. And really our philosophy for all of this is to be designed to be as flexible and have as broad an offer as is possible. Some of you are going to have different needs. Some will require guide RNA design with validation. Others are just going to be looking for a backbone to insert the guides you've already designed. Some will require high-level scientific support from an ongoing basis. Others will require donors. So all of these products and services, we have designed them to be able to meet your needs, whatever they may be. So the ones that are in blue are the ones that are currently available and on the market right now. The ones that are in red at the bottom are ones that are coming soon. So I'll just take a moment to speak about each of these. First off, Cas9. As Eric has mentioned, there are 2 different varieties of that, the wild type and the nickase, and we have both of those available right now. As a plasmid, they're available separate, so just the Cas9 and the plasmid, or in combination with a guide. I'll speak about the guides a little bit more in a moment. And they're available in two different volumes. A cloning volume, which is most appropriate when you have to do some sort of modification to its cloning in a guide, for example, and that's 1 microgram. Or a larger volume, which is 5 micrograms, which is transfection ready. So it's a high enough volume, so it's ready to use straight out of the material once we send it to you. It's also worth taking a moment to pause and state our philosophy around CRISPR from an IP perspective here. We're dedicated as an organization to making sure that our customers who work with us are going to have freedom to operate going forward in the CRISPR space. We are active in the space. You may have already seen we've done a press release on one piece of IP. We're busy in other areas as well, which I can't get into detail at the moment, but that is an important platform for our process and what we want to offer to our customers. On the guide RNA side, again, the plasmids are available both separate, so you can clone in yourself, or combined with Cas9. And available as single or double guides. So as Eric mentioned, when you're using the nickase version of Cas9, having the dual guide system is extremely efficient and specific. So we have both of them, and they can be put on the same plasmid. Available off the shelf, and also custom guide generation is available with validation. I'll cover that a bit more in a moment. Jumping down to the bottom right, we are working on some cell lines, a couple of different versions of them that are worth noting. The first is a CRISPR-ready cell line. So this is one that is already being genomically modified to be constitutively making the nickase version of Cas9. Our in-house work has suggested that the wild-type version of Cas9 can be toxic, but the nickase version is well tolerated by the cells we've worked with thus far. So that's a great starting point, a leapfrogging point for any further modifications. So by using a CRISPR-ready cell line, you can modify it further, and all you need to do is get the guide inside. The landing pad cell line is one that has a LOXP site, and potentially one of either just the LOXP by itself, or one of 3 different promoters in front of varying strengths. That's more of an overexpression system. And finally, for the cell line side, our entire menu of over 500 different off-the-shelf cell lines are available for further modification. So if you wanted, for an example, a KRAS G12D cell line, then you wanted to do a further modification in BRAF, then we're absolutely well positioned to be able to support you in that, almost uniquely positioned in the marketplace, I would say. Now, if the CRISPR-ready and landing pad cell lines you're interested in are not ones that are available off the shelf right now, which, to be honest, is most of the time. There are a lot of different cellular backgrounds out there, and we can't develop them all. We have the do-it-yourself platform generation, cell line generation kits. These are the plasmids that we have used to generate the CRISPR-ready and landing pad cell lines, and they're available to you as a kit, so you can make your own CRISPR-ready or own landing pad cell line in your background of choice. Down to the bottom left, we have donors that are available. We have backbones that are available for your own cloning. We also have them available in both plasmid and rAAV format. And we have, as Eric was mentioning, the cluster donor generation process. And those are available either as a report, as an oligo, or cloned into a stable vector. And finally, there's the services, which we have available of viral encapsulation, not just of the AAV, but of really of any of the plasmids. Ongoing project design and support, and troubleshooting. So the off-the-shelf plasmids that are currently available, and I'd say watch this space, there'll be a lot more coming in the very, very near future, but this is what's currently on the marketplace. What we have is we've got guide RNA plasmids, both available for the single or the dual in all-in-one vectors, and that's the GE6 and GE9 that you see at the top. So that's with the Cas9 present. And also then the GE12 and GE13, which does not have the Cas9, that's just for the guide. So if you want to do dosing experiments with different amounts of Cas9 present along with a specific guide, we can meet your needs. We have the
Sorry, I just wanted to point out, sorry to interrupt, Chris, too, that in the graphic there, I think the Cas9s are not distinguished from wild-type and nickase version, but in fact, with a single guide, you're using the wild-type. With the dual guide, you're using the nickase version.
Thanks, Eric. Then there's Cas9 plasmids themselves. So we got wild-type in 2 different volumes. Again, as I mentioned before, we have the cloning volume and the transfection volumes, and the nickase, again, in both the cloning and the transfection volumes. So for the guide RNA service, this schematic at the top describes really what the offer is. First off, once we've worked with you to identify what the target is, we'll do in silico guide design. We'll look at a significant number of different guides in order to determine what the right ones would be. We'll generate a report is the first option. Actually, in every case, no matter how far you proceed, we'll generate that report with our predictions of what the cutting efficiencies will be. If you choose to stop at that point, that's perfectly fine if you want to do your own guide manufacturing and cloning. So we can either provide just the reports, or we can provide the reports plus oligo. If you'd like us to do the cloning as well, we can do the cloning into an all-in-one vector. Now that's our default. If you'd like us to clone into any of the other vectors that we have available, more than happy to do that. And then we provide the report and the clone. If you want more than one clone, as for example, some people may just confirm that even though we predicted in the in silico design, everything seems like it should be working fine. If you would like more than one of these clones, we can make those available. Another option is to actually have it packaged into adenovirus, which is ideal for hard-to-transfect cells, such as iPS cells or certain neuronal or cardiac cells. So we're very happy to provide that. And then finally, there's the guide validation. In the guide validation, we take a look at 5 different guides, confirm through the surveyor assay, as Eric has described, and then clone the highest performing of those. We do this in K562 cells, so we do them in human cell lines, but we're adding mouse very, very soon, and potentially other species as well, depending on demand from the marketplace. As I mentioned, the quick start cell lines, what we have right now are 2 CRISPR-ready cell lines that will be available very soon. In MCF10A cell lines, we've got CMV Cas9 nickase, and then HEC293 cell lines with either PGK or CMV promoters, again Cas9 nickase. And again, as I mentioned, when a background is not available in the cell line of interest, we provide these kits that allow you to generate 5 different CRISPR-ready cell lines off of the plasmid material. And we have landing pad cell lines coming soon in either just the LOXP or a couple of different promoters. To pull all of this together into how these models can be used to generate the cell lines you want, if you're looking to do an overexpression model, the best place to start is a landing pad cell line, and if we don't have it, the Quick Start Landing Pad Development Kit is the tool that's the right one for you. If you're looking for an endogenous system, you can either start with a CRISPR-ready cell line if it's available, or develop your own. Again, the perfect model if you want to develop a significant number of genomic modifications off of a single cellular background, or use any of our off-the-shelf cell lines. That'll ultimately drive you towards your targeted cell line. And just a word on service and support. To sum this up, as Eric has mentioned, gene editing still isn't easy. Perceptions are that it is easy, but at the same time, what we're seeing, and from speaking to a significant number of our customers, there's some stumbles that can be made along the way, and we'd like to help you get through them. Our scientists use CRISPR and rAAV every day. We are experts in the space. We have a large number of scientists who are actively improving the technology on an ongoing basis, regularly using it on the behalf of our customers and for our own internal development programs. So a very, very strong group that we're happy to help you with, and we're actually offering you access to that expert support, really to reduce the risk of failure and the risk of lost resources, time, and money. And so it can be used for upfront project planning and project support and troubleshooting. And one other thing I'd like to mention is that because Horizon has a very broad menu of cell lines that are available, we are very actively interested in licensing back any cell lines that you develop. So in terms for a royalty and also for genomic validation of the cell lines that you provide us, we'd be very interested in working with you on anything that you put together.